Hi, this is Miss Slitton, and this is our little tiny crew of ladies that are here to review Chapter 7, Cellular Respiration. We're super excited to be here. Say hi.
Hi.
All right. Now, big picture. This is how cells metabolize nutrients, and they transform the energy, the potential energy that's in the food, into ATP. That's ultimately what we're trying to do. And this is augmented if we have oxygen gas present, and then we're doing something called aerobic respiration. If we don't have oxygen present or not enough oxygen, we do anaerobic respiration, which we know as fermentation. These happen in different places in the cell, so this is a good overview now that you've learned this already, and it's a review for you. All cells will do glycolysis, and they will have that occur in their cytoplasm. Is this a picture of a eukaryotic cell or a prokaryotic cell? Eukaryotic cell. How do you know it's a eukaryotic cell? It has membrane-bound organelles. They're so smart. And so I can see the mighty mitochondria in there. Now, prokaryotic cells can do this too. They have membranes that they can do their electron transport chains on. They're just not isolated like it is here in the mitochondria. Okay? So anyway, so we do glycolysis in the cytoplasm, and when you're all done with glycolysis, we know that you reduce somewhat NAD and you make some ATP. How many? 2. And if you don't have any oxygen, those are the only 2 ATP you're getting from your glucose. But if you have oxygen, you can do, what's the next step? Transition prep, and you can transition right into the mitochondria and convert your end product from glycolysis. What do we get? 2 pyruvic acids. You can take those 2 pyruvic acids and can continue to oxidize them into 2 what? Acetyl-CoA. And we will, but one way, because we're going to feed each of those acetyl-CoA through a Krebs cycle. Now, in the process of transition prep, we will again write some more checks, right? And our checks in this case are reduced NAD. Right, so we take our 2 pyruvic acids, and when we're oxidizing our pyruvic acids into acetyl-CoA, we're making 2 more reduced NADs. When you do the Krebs cycle, you're spitting off all kinds of carbon dioxide and making more reduced NADs and FADs, and so that when you go to your last step, your electron transport chain, you have a source of powerful electrons. Your NADs and FADs have high energy electrons that they drop off at the top of the electron transport chain. And as those electrons pass from one receptor to another on that electron transport chain, if you come in at the top, there's 3 places where you can push hydrogen ions from the matrix to the intermembrane space. So we know the matrix is here, right? And then the intermembrane space is here. And at 3 spots along that electron transport chain, we are, do you remember what those are? Reductase, cytochrome oxidase. Good. You're my APers in here know this. You're concentrating all this hydrogen right here, so when it moves back into the matrix, and it's going to move back in using the ATP, what? Synthase complex. Good. You can generate a bunch of ATP. Now that ATP will then diffuse out of the mitochondria. It can be used in any kind of metabolic reactions that you have in that cell. That's what the energy currency is of that cell. So basically what we did in class is talk about what are each of those steps so we could understand it better. So let's just go to the Minecraft version of glycolysis. And we know it's getting shown as one single reaction, but we all know how many steps is it actually? Yeah, it's 10 steps in here using what? 10 different enzymes, exactly. And ultimately, you're taking this glucose that you have right here, and you're basically just breaking it in half, right? And we know that glucose has 6 carbons, and each of these pyruvic acid has 3 carbons, and since I have 2 of them, 2 times 3 is what? 6. And we know that glucose has 12 hydrogens, but 2 times 4 is what? 8. So I have lost some hydrogens. the hydrogens and their energy, their electrons, were given to who? NAD. That's how it got reduced. It got reduced with the hydrogens from glucose. And basically, you're repeating that process again and again through transition and through the Krebs cycle. You're taking away the energy and in the hydrogens, and you're doing a reduction to NAD or FAD and giving it temporarily to those two molecules. Then they will give up that energy at the top of the ETC, the electron transport chain. So when you're done with glycolysis, you'll walk away with 2 NADs and 2 ATPs. But is that the only ATP I made in glycolysis? Do you remember how many ATP I actually made? That's right, I made 4. But I had to spin 2 ATPs to get this ball rolling, to get this party started. That's why I walk away with 2 ATPs. Okay, so this is showing you inputs and outputs. Ask your bio buddy that you're sitting next to, and all my AP biologists over here and there, do you have any problems with those inputs and those outputs? Do you understand where they're all coming from? Why is ADP an input? Because I need some ADP to make ATP, right? Because I'm going to take an ADP, I'm going to add an inorganic phosphate to it so I can make an ATP right here. Yes? And that's making ATP without using any electron transport chain. It's just a handoff. I have the phosphate, now you have the phosphate. That's called substrate level phosphorylation. No ETC needed. Pure handoff. Pure handoff we also see in the Krebs cycle, right? 2 + 4 is 6, do the bit, get 5, do the bit, get 4, make an ATP. That's no electron transport chain needed. Okay. And now that you have studied photosynthesis, that's called photophosphorylation because you're making ATP using energy from the sun to make your ATP. Good. All right, so once we're done with glycolysis, if we have oxygen, we're going to mosey on in to the Krebs cycle. This mosey process, we will make 2 NAD that are reduced here. And that's what we call transition prep. And then we will do our Krebs cycle. We will have 2 acetyl-CoA. These are 2 carbon units. And we're gonna, what's already preloaded in the Krebs cycle? A 4 carbon, right? And that's why we say 2 + 4 is 6. We make a C6. We do the bit. We pull off a CO2, right? What else do we do? Reduce an NAD, right? And we get a C5. We do the bit again, and we get a C4. Make an ATP, reduce an FAD, reduce an NAD, and we bring in our second acetyl-CoA that we made. Yes? This picture right here, without the stuff on it? With the stuff on it? Okay.
Does the NAD get reduced by the oxygen and the NAD at first? Like, okay.
Oh, oxygen.
CO2. The oxygen come from the NAD?
Wait, say that again?
So, you know how CO2, the oxygen comes from the NAD, that's what makes it reduced?
Oh, are you doing it for her?
Yeah.
Oh, okay, gotcha. Wait, I'm so sorry.
Okay.
For the 3rd time, I promise I'm listening.
The carbon comes from, you know those, yeah. But the oxygen comes from the NAD, right? That's how you reduce it.
You don't need any oxygen to do that. Remember, there's different ways to reduce something. You can give it electrons, you can give it energy, or you can take away oxygen. All those things will reduce. We're giving it energy by giving it electrons. We're giving it that hydrogen. Not by taking away oxygen. Good question, though. Do you have your picture? I was walking right in the middle of it. Okay, so then what you do is you take all your reduced NADs and FADs and you take them to the top of the ETC. That's not actually true. Why? Because it's a lie. Because do all of them get to go in at the top? No, some come in part way, right? Who comes in part way? Oh, the FADs. That's the AMP check, right? So they don't get the first reductase. They get the second reductase cytochrome oxidase. They only pump out 2 hydrogens, so they only get 2 ATP. All right, so here we can see the structure of the mitochondria that we already talked about. And as far as where does everything happen, right? Glycolysis occurs out here. I need something lighter. Transition prep happens inside the mitochondria. The Krebs cycle happens inside the mitochondria. And then the electron transport chain, right, is happening on the electron transport chain is happening on that inner membrane, right? Okay, and it's pumping hydrogen ions out into this inner membrane space right here, so that when the hydrogen ions go back in, that's when they make the what? ATP. All right? So then what we did is we looked at transition, right? And then we even looked at it in better detail over here. Remember this part? We said, look, here are the 3 carbons in pyruvic acid, and that came off as a carbon off each one. Here it is right here, because there's only 2 carbons here. And then we looked at, here's the CoA. Well, the CoA is sitting here. It took that carbon spot. And then the hydrogens, which are here, went here, right? And then we could see the CO2 that's here is the CO2 that's there. And that's why we went when we came across. Because we pulled off the carbons as carbon dioxide, we docked in an acetyl-CoA. All right? Clarifying questions on that part?
This is aerobic respiration, right?
We haven't even got aerobic yet.
Okay.
Now, the thing is, when you do glycolysis, you do transition prep, you're going to do the Krebs cycle, and then you're going to do the electron transport chain. This is when you need the oxygen, and it's going to form the water, not till here. But if you don't have the oxygen here, no electron transport chain, no Krebs cycle, no transition prep, and instead you're going to go down the fermentation pathway where you're either making lactic acid or alcohol. Yeah, it makes our muscles sore. Is that good? So you really don't need it to the very end, but it backs everything else up when you don't have it. All right, so then we went into, we didn't like that one, right? We like this one, right? And so then when this is in our song, we say 2 + 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and 2 + 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and stop. Then we gathered all our reduced NAD and FAD, and we take it to the top of our ETC, and then right? So if you come in here at the top, you get reductase, cytochrome oxidase, right? Then you make some ATP. All of these 3 come back in. A lot of it, a lot of it, a lot of it. But if you only come in here, right here, then you only get this one and this one, and a lot of it, a lot of it. Only 2 come back in. Okay, and then who catches the electrons at the bottom of our chain? Because they've been passed along here. Somebody, if we want this to keep going, it's like an assembly chain. We need to keep removing electrons if we want it to keep going. So who keeps catching these electrons at the bottom? Oxygen forming water. And because we cranked out all these hydrogens out here, this is why we get to make so much ATP. A lot of it over here. I don't know what I just did. Okay? But if we don't have oxygen, then we're going to do our glycolysis. This right here is glycolysis. That part's normal, happens in the cytoplasm. We'll reduce some NAD. But now, since we can't do transition prep, we can't go in the mitochondria, so we're going to do fermentation. How many choices do we have? 2. You and I make lactic acid, makes our muscles sore. Plants and yeast make alcohol. Either way, though, what happens is the NAD that we made during glycolysis, the reduced NAD, ends up getting oxidized again. Why is that important? So we can reduce it again. And then we do fermentation and we oxidize it, and reduce and oxidize, so because we have a finite amount of it. All right, got it? Want to do the whole song? Join with us at home, though you can't see us right now. Ready? What's the whole thing called? Cellular respiration. What's the first step? Glycolysis. Who does it? All cells. So cute. Where do you do it? In the cytoplasm. What do you start out with? Glucose. How many carbons? 6. What are we going to do? Break it in half. How many steps? 10. How many enzymes? 10. What are these called? 2 pyruvic acids. And you reduce some NAD and make some ATP. 2. Oh, we have plenty of oxygen. What should we do? Aerobic respiration. What's the next step called? Transition prep. Here we go. And you reduce some NAD and 2 acetyl-CoA, put 1 away. What's the next step called? Krebs cycle. What's the name for it? Citric acid cycle. How many times are we going to do it? Twice. Why? 2 acetyl-CoAs. And every time we do the bit, what are we doing? We're reducing an NAD and pulling off a carbon dioxide. And here we go. 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and stop. Gather all our reduced NADs and FADs and take them to the top of the ETC. Reductase, reductase, cytochrome oxidase, and you make some ATP. A lot of it, a lot of it, a lot of it, or a lot of it, a lot of it. Who catches electrons at the bottom? Oxygen forming water. Water, 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 water. What are we going to do? Break it in half. What do we get? Oxygen and electrons. Sweep, sweep. They don't know this yet. Here comes the sun traveling in waves. Particles of light are called photons. And it gets the electrons excited. PQ. And you make some ATP. Here comes the sun traveling in waves. Particles of light are called photons. And it gets the electrons excited. Who catches it? NADP forming reduced NADP. Then you take some ATP and some reduced NADP to the dark reaction, or the Calvin cycle, or the light-independent reaction. 3C5 plus 3C1 makes 3C6. Break down to 6C3. This is photosynthesis. Use some ATP. Use some reduced NADP on your 6C3. Take 1 away, PJL, jazz hands, and you're left with 5C3. Use some ATP to build 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. Take 1 away, glucose, jazz hands, and you're left with 5C3, use some ATP to build 3C5. Stop. Now pick up your glucose. How many carbons in it? 6. What are you going to do? Break it in half. Steps? 10. Enzymes? 10. What are these called? 2 pyruvic acids. And you reduce some NAD and make some ATP. 2. Oh no, we don't have any oxygen. What are we going to do? Fermentation. How many choices do we have? 2. You and I make lactic acid. Makes our muscles sore. Plants and yeast make ethanol. Oh, sorry, alcohol. Either way, you oxidize your NAD. Yay! So you had a photosynthesis song put in there because the song's link. So that's coming up. Those of you studying for the quiz, don't freak out about the photosynthesis song. I will teach you that next week, or you can watch it on this YouTube channel. Okay, thanks AP for helping.